Hello, my name is Jason Vanstone. I'm a research scientist with the Stewardship and Clinical Appropriateness Department with the Saskatchewan Health Authority. I work with our antimicrobial stewardship program. This video is going to be a general overview of what antimicrobial stewardship is and why it's an important program to have within a health authority. So I'll start with the basics really, which are talking about bacteria. We need to understand what they are before we can really delve into what antimicrobial stewardship is. So bacteria are single-celled living organisms, and as such, they have several features that make them a living organism. So for example, they have to be able to protect themselves from the outside world, protect all the good stuff inside from what's on the outside. And so to do that, bacteria have several different things. They might have a capsule, this red layer that you see on the outside of the cell here. They'll have cell walls, which is this yellow layer, and then this green layer inside is the plasma membrane. That's something that's common to all living things. But what these are basically is physical protection, keeping the stuff on the outside from the stuff on the inside. Cells also have to be able to get around, and so these pili here are something that bacteria might use for movement. They also have a flagellum here that you can see, kind of like a propeller, and that thing will whip around and helps these bacteria move around so they can search out food and places to live in their environment. And cells also have to be able to grow, create the proteins that they need to be able to grow, and in the case of bacterial cells, divide and create new cells. And so living things do that with ribosomes. These are these little blue spherical structures you can see dotted around the cell here. And you can think of these like living machines, basically, or protein building machines within the living cell. And so they take the information that's stored in the DNA and they translate that into proteins. And the DNA in a bacterial cell here, you can see is there's this big red squiggly thing in the middle. That's the bacterial DNA. So they're big circular kind of single chromosome that they have. And lots of bacteria also have these plasmids, these extra little chromosomal pieces of DNA. And so those are the important pieces for sharing and storing information. So that's what, again, common to all living things. They have to be able to share and store their information in order to divide and reproduce. And so that's done in the form of DNA. And we'll talk more about these plasmids later. They're actually a very important piece for sharing antibiotic resistant genes in bacteria. So these are the basics of a living thing and the basics of a bacteria. And just to touch on here that there are lots and lots and lots of different bacteria. These are very ancient creatures. And so they've evolved many, many different forms and they've spread across the entirety of planet Earth. And so the ones that I'm showing here on the screen are actually all disease-causing bacteria. See, there are lots of different ones. You might recognize some of them just by their names. But I do want to highlight that not all bacteria are bad per se. They're not all disease-causing. And so there's something called the human microbiome, which some people may be familiar with. And this is essentially all of the bacteria that live in or on our human bodies. So this little pictograph here shows sort of the ratio of human cells in our body to microbial cells. So a human body is basically about 30 trillion cells. That's what makes us up as individuals. But there are about 100 trillion bacterial cells living in or on us. So if you think about a human being on a cell-to-cell ratio, we're actually about 3 times as many bacterial cells, excuse me, as we are human cells. And so it's just a very interesting concept when you think about that in terms of, what are we? But because of that, these bacterial cells play very important roles in human health. And these are things that we've known and really been studying for several decades, but we're still starting to learn some of the consequences of messing around with our human biome. So you can see this figure here just shows some of the different areas that the human microbiome interacts with the human body and creates sort of an interaction between our environment and our body. And some of the different organ systems and various areas of the body that these bacteria provide beneficial effects. And a lot of them will be things like assisting digestion, creating, helping with safety and preventing pathogenic, those bad bacteria from getting a foothold and infecting us. So to compare now the bacteria to viruses, because these are different things biologically speaking. So there are lots of different viruses, just as there are bacteria. But there are some important differences. And so you can see here, I've got a number of different viruses laid out. And for the most part, the pictures above kind of show what they are. And it's essentially a protein shell that contains the DNA or the RNA, whatever nucleic acid they use. And so often they're spherical shape, but they can be different shapes. Here's an example of Ebola virus, which is this long, squiggly, spaghetti-like thing. But they're essentially that simple. These are DNA inside a protein shell. And so the way that viruses work is one of the things that makes them very different from bacteria. And as we'll come to see, what makes them resistant or what makes antibiotics ineffective against viruses. So here, if you look at this big brown thing, we could imagine that's one of the cells lining our airways in our lungs. Let's say this viral particle over here is a cold virus, something like that, that's coming in to infect our body. So the virus comes in, it gets inside of our cell, and so you can see there's that protein shell and then the DNA inside the virus. What happens is the virus opens up and releases that DNA, and it goes into our cell's nucleus, and it will trick our body, our cells, into using our cellular machinery to make more copies of the DNA. So it makes more copies of the viral DNA using our cellular machinery. It uses our cells' ribosomes to then make the proteins from that virus. And then the virus is able to rebuild itself and spread off to other cells, and it'll have multiple copies of the virus that can spread out that way. So this is how viruses work. It's very different from bacteria, in which all of this replication is built into one living cell. And so that's kind of what, sometimes we'll when we say viral life cycle, viruses kind of in the biological sense, they're not really living because they're not able to replicate their own DNA, to make their own proteins, and to replicate themselves. They require a host to do that for them. Okay, so just to kind of give a big broad background about the importance of infectious disease, so viruses and bacteria, in terms of sort of human history and overall human health. If we look back to about 100 years ago, in the early 1900s, about 46% of the deaths from in the United States were from infectious diseases. And we can see that has changed down to about 3% in 2010, just over 100 years. And so that's really a huge change to go from almost half of the people who died dying from an infectious disease to only about 3%. That is a huge shift in that time frame in terms of what people are dying from. And if we overlay a life expectancy graph on there as well, we can see that in that 100-year span, average life expectancy went from about 45 years in the early 1900s to about 80 years currently in most countries. And so, again, a huge shift, not just in what people are dying from, but how long we're able to live. And of course, there have been a lot of advancements over that century in terms of medicine and medical science. But specifically talking about infectious disease, one of the key components of that change that we're seeing is the discovery of antibiotics. And so this is Dr. Alexander Fleming, who was a Scottish physician working in the UK in the early 1900s. And he was a bacteriologist by training, so he studied bacteria. And this is an example of a plate that one might have seen in his lab. So it's got this kind of white, creamy looking stuff kind of across the petri dish here. This is the bacteria growing on here. And so what happened was, that sometimes happens in the lab, you'll get contamination on your plates. And so Dr. Fleming came back to his lab and discovered that there was a fungal colony, this big white fluffy looking thing here, growing on one of his plates. And rather than tossing it aside, he really took an interest in and really wanted to look into this phenomenon that was seen here, where you have this zone of inhibition, this area where the bacteria are not able to grow right up against that fungal colony. So he wanted to know what was going on here. So he looked into this, and over years and lots of experiments, was able to identify that the fungus here, which is a penicillium fungus, is actually releasing a chemical into the environment, which we termed penicillin. And that is actually an antibiotic, as we call them. So we kind of stumbled upon this chemical warfare that microorganisms use to compete with each other. So in the world of living things and natural selection, all things have to compete for resources, for food, for space to live, things like that. And so it's no different in the microbial world. But because they don't have teeth and claws and nails and stuff to fight each other with, they use chemicals. So the fungus releases this chemical, this antibiotic, to kill off bacteria in the area around it, which will allow the fungus space to grow and use all the food and resources in that area. So we really stumbled upon this warfare, and were able to kind of weaponize it for human use. So we were able to turn these into drugs, and that caused this explosion throughout the rest of the 20th century. So that was in 1927, I believe, when Fleming made that first discovery, and penicillin really first made it into market in the 1940s. So you can see there's just discoveries of different classes of antibiotics as we sort of go through the mid-1900s there. That all work in different ways to kill different types of bacteria. So let's talk a bit about that. How do antibiotics work? Well, some antibiotics might work, and there's just some examples there, by disrupting the cell wall, either inhibiting its synthesis or maybe depolarizing the plasma membrane, so they kind of punch holes in it, which causes changes in the water inside the cells, and they can explode cells from the inside. You might have antibiotics that work by inhibiting protein synthesis. So they will mess around with the ribosomes to prevent them from being able to build those proteins and do what the cell needs to live that way. You can also inhibit DNA replication with some antibiotics. So they'll actually prevent the cell from being able to copy and store that DNA for the new generations of cells. So if you think about back to that first slide, antibiotics work by breaking down these key pieces of living things, being able to protect themselves, being able to grow, and being able to share and store that information. And so I'll just take a moment to highlight here, this is why antibiotics don't work on viruses. Viruses don't have these cellular structures. They don't have ribosomes. They don't create their own proteins and cell walls and things like that. And they don't copy their own DNA. They use our cells to do that. So if you have a patient or someone that has a bacterial viral infection, like a cold or a flu or something like that as an example, giving them an antibiotic actually provides no benefit to that patient. And in fact, only exposes that patient to the potential harms of antibiotics. And we'll talk about that later. Okay, so let's talk a bit about antibiotic resistance, because that's really the big issue that is facing people in medicine right now as far as antibiotic use goes. So when you have your regular run-of-the-mill bacteria and you use antibiotics against them, they will die. And that's great. That's the whole purpose of having these antibiotics. And so you can see in these regular bacteria, there's their chromosome, and they might have this extra plasmid. And the difference when we talk about antibiotic resistance is only that these bacteria have somehow found their way to developing or acquiring antibiotic resistance genes. So this is just a genetic change that now these bacteria are able to resist the effects of antibiotics, and those antibiotics become less or ineffective at killing the bacteria that they once used to be able to kill. So what does that mean when we talk about this? What are these mechanisms? Well, again, just hypothetically speaking, here's again your bacteria, and maybe, for example, that bacteria acquires this green gene right here, and this creates a drug inactivating enzyme. So basically, when you get the drug, which is these blue spheres in this case, when these come into the cell, this enzyme can go over and chew up that antibiotic and get rid of it before it's able to kill the cell. Or maybe here's a gene that's found on a plasmid that might create a modified cell wall protein, which actually stops the drug from being taken up by the cell in the first place. I think I had another example. Yeah, the purple one here. So this gene, maybe that creates a drug efflux pump or activates an efflux pump. So as soon as the antibiotics get inside the cell, this efflux pump pumps them right back out before they're able to have any bad effects on the bacteria. And so one of the issues with antibiotic resistance is that it spreads quite easily throughout bacterial populations. And there's lots of different mechanisms for doing that. One thing I want to highlight is just how we can actually view this in almost real time. So what's showing here on the screen is an experiment that was done at Harvard a couple of years ago. And what they have is this is a giant petri dish, basically like the size of kind of a table. And on either end, in the medium, they had increasing concentration of antibiotic as it goes towards the middle. So you can see along the bottom there, there is no antibiotic in this outer section. And then as you move in, there's a 1 times and a 10 times and a 100 times concentration of antibiotics, so that by the time you get to the middle portion of this giant petri dish, there's 1,000 times more antibiotic than there is out on the outer edges. So they plated these things with bacteria. They put the bacteria onto them, and they let them grow. And they were taking time-lapse pictures, so you can see these bacteria starting to spread into the different areas of the plate. And what's interesting is you'll note that both of the outer edges are covered in this lawn, this carpet of bacteria, because there's no antibiotics stopping them from going in. But then they get to that kind of invisible barrier where there's all of a sudden there's an antibiotic in that media. And so now only the bacteria that are resistant to that antibiotic are able to keep growing. And then similarly, they'll grow up to that next edge until there's an increased concentration of antibiotic. And so now only bacteria that are able to resist that higher concentration of antibiotic are able to grow into that next area. So I'll just speed this up a little bit. And you can see these things sort of starting at a point and then fanning out as they grow further and further towards the center of the plate. So just to kind of overlay an evolutionary tree on that, if you will, you can see, and you can actually trace back some of these bacteria from the middle here. You can trace their lineages back through these kind of fan-like explosions of growth, all the way back to their sort of great grandparents, all the way back here. But the bottom line is the bacteria that are living here, we've been able to watch them evolve so that they are now basically 1,000 times more resistant to antibiotic than their ancient relatives all the way back here. So we've been able to watch this happen in real time. We've seen bacteria go from having no resistance to developing and increasing that antibiotic resistance as we watch this plate develop. And so, this is one of the issues with bacteria is they grow fast. And if you think about E. coli, for example, which happen to grow, there are bacteria that can grow inside of us, and they happen to grow best at 37 degrees Celsius, which is human body temperature. And if that happens, I think it's about once every 20 minutes, E. coli can produce a new generation. So one bacteria becomes two become four become eight, and then you can see just like on the GIF that's going here, this explosion of growth of bacteria in a relatively short period of time. So even one resistant bacterial cell can become many, 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 many bacterial cells that have resistance over a short period of time. And of course, one of the neat things from a biological perspective of bacteria is they've got lots of different ways that they can share genes. And so here's one of them. This is example in the picture here. This is called bacterial transformation. And so what this is that bacteria actually have the ability to take up DNA from their environment. So what we've got on the left here is a donor cell, and this bacteria had antibiotic resistance, a gene for antibiotic resistance, and it dies and it kind of breaks apart. You can see the cells kind of exploding here and it kind of disintegrates, but the bacterial cells nearby are able to kind of slurp up some of that bacteria. And if they end up taking up one of those antibiotic resistant genes, then the recipient cell can now become antibiotic resistant without actually having to go through the process of evolving it on its own, that trial and error of creating new daughter cells and evolving antibiotic resistance genes. It can just pick them up from the environment where other cells have lived and died. Another way that bacteria can get these antibiotic resistant genes is through a process called transduction. And so this is what's showing here is this little kind of spaceship looking thing is called a bacteriophage. And so these are bacteria that's viruses that specifically infect bacteria. And so what happens, similar to bacteria that infect humans, when they repackage that virus inside of the infected bacteria, sometimes they might actually get a little piece of the bacterial DNA repackaged with that viral DNA. And so if it ends up taking a piece of DNA that is an antibiotic resistance gene, that can get packaged into that virus. And then when it goes off and infects another bacteria, it can release that antibiotic resistance gene into that bacterial cell. And now again, you've got a bacteria that has been able to achieve getting antibiotic resistance without having to evolve it on its own. The last way that we can talk about bacteria getting resistance genes is through bacterial conjugation. So this is where these plasmids will come into play again. And so this is kind of the equivalent of bacterial sex, where one donor cell will create a connection between it and its recipient cell, and it's able to transfer plasmids through that connection. And then that, again, confers antibiotic resistance to the recipient cell. The neat thing here too is that we've seen that not only can this happen between members of the same species, but it can actually happen between different species of bacteria. So you can get these plasmids that go from one bacterial species to another bacterial species, which creates even further complications for trying to control and manage antibiotic resistance once it's in a bacterial population. Okay, so that's kind of antibiotic resistance in a nutshell. Well, what does that mean for people? So when we look inside of people, and we talked about the human microbiome, so we've got all kinds of bacteria that live inside of us or on us. And so here's just an example looking inside of a person. They might have, this kind of population of bacteria growing inside of them. And maybe they're one of those unlucky people that's got some antibiotic resistant bacteria already in there, these guys with the plasmid that you can see. But for the most part, because this extra little bit of DNA takes a little bit extra in terms of energy to allow these cells to reproduce, then for the most part, these cells are outcompeted by all of those beneficial bacteria that exist, in this case, within the human gut. So they're not able to really get a foothold and take over and cause any real big problems. The issue comes when people take antibiotics, and especially if you're taking antibiotics unnecessarily. So you take those antibiotics, it goes into our stomach, it is absorbed, and it spreads everywhere throughout the body. And the antibiotics aren't targeted, so they will kill anything that is not resistant to them. So that's what will happen here. We get killing off of these things. Now, if you're treating an infection, that's fine. That's one of the things we want to do, is to be able to kill off those bacteria. But if you're not treating an infection particularly, you take your antibiotics, and then the trouble happens at the end of your treatment. So once you're done taking those antibiotics, you've eliminated a bunch of your good bacteria, and now potentially what's left are these ones that might cause more issues, these antibiotic resistant guys. And now they don't have anything competing with them. And so they get a chance to get a foothold and grow and reproduce. And now you might have a problem. Now you might be colonized with these resistant bacteria. And in fact, this might cause more problems the next time you have an infection. And they, excuse me, in fact, might be the source of a next infection. So hopefully this emphasizes why it's important to take antibiotics only when they're necessary. This is something that every single course of antibiotics can drive this antibiotic resistance. And this doesn't even consider some of the other side effects of these medications. Antibiotics are no different than any other medication in that they have side effects. They are not benign. And so they're not something that should be used sort of just in case, because they can have serious side effects. So what are the consequences of us not changing our behavior? We know that we use a lot of antibiotics unnecessarily. We know that this is a problem everywhere in the world. And so what happens if we don't change what we're doing? And so there was a review done back in 2014, and they showed that if we don't make changes to our behavior in terms of our antibiotic usage, we could see by the year 2050 more than 10 million deaths each year due to antibiotic resistance. And so you can see that that's not a burden shared equally around the world. There are certainly places where those numbers are much higher than others. But to put that also in context of what that 10 million means. So currently, we see about 8 million deaths a year from cancer. That's this big circle on the bottom right here. Whereas we only see about 700,000 or about 1 million deaths a year related to antimicrobial resistance. If we don't do anything to change the way we use antibiotics, we could see that number explode to 10 million by 2050, which would be more than we currently see in terms of people dying from cancer. And so that's kind of a scary and sobering thought to think that we could essentially be going back towards the dark ages of medicine, this time before we had antibiotics. And so if you think about that graph that was showed at the beginning with that switch from 46% of people dying from infectious diseases in the early 1900s down to 3%, this represents potentially seeing that start going back the other way, where we're going to see more and more people dying from what are currently preventable illnesses, things that we can treat with antibiotics. The more antibiotic resistance we see, the less likely we are to be able to treat all of those with our current antibiotics that we have. And so the last piece I want to touch on is that I've obviously been talking a lot about this as a human health issue, but I want to make the point here that antimicrobial stewardship itself is not just an issue of human health. This is something that involves multiple sectors. And so to exemplify that here, there's a little graphic. So this is us, people living in our community. And George is the guy in the center here. And so George gets sick and he gets antibiotics, whether it's appropriate or not. And he ends up developing resistant bacteria in his gut, unfortunately. Now, maybe George stays at home because he's not that sick, and he's just out and about in the general community. George isn't a great hand washer, and so he's out getting groceries and picking up apples and touching door handles and things like that. And he spreads his resistant bacteria. Now that's picked up by other people in the community who are touching those same surfaces or shaking hands with George, whatever the case may be. Another scenario there is maybe that George is actually quite sick and he gets care at a hospital or he's in a nursing home or another inpatient care facility. And the resistant bacteria are able to spread from him to other patients and healthcare providers within that healthcare facility. And then in a similar fashion, those patients might go home, back to the community. The healthcare workers might, they head home to their residences at the end of the day. So we're able to see that antibiotic resistance spread through that route. Another potential spread is directly spreading resistant germs from between patients. So George is sharing a room with someone and spreading through shaking that person's hand or whatever. Again, we know George isn't a great hand washer, so or even spreading person to person through the healthcare providers. Hand washing is a big thing for healthcare providers as well. So you can see that there's these multiple ways that these antibiotic-resistant bacteria can be spread, and that's just within sort of the human sector. So now if we consider the animal world, so food animals, we use antibiotics for again, multiple reasons to treat these guys. And then same thing, they can also develop resistant bacteria in their guts. During the butchering, those drug-resistant bacteria can contaminate the meat and remain on those products. And when they're not handled or cooked properly, then those bacteria, antibiotic-resistant bacteria, can spread to humans. And then through fertilizing or water containing animal that's contaminated by animal feces and these drug resistant bacteria therein, they might get used on food crops. Those drug resistant bacteria can remain on the crops and be eaten and then get into the human gut. So again, you can see these cycles and these paths through which antibiotic resistant bacteria can spread. And so the point here is that this One Health idea is that human health and animal health and environmental health are all connected. So this One Health is at the center of that. And so we need to understand that when we're going to practice antimicrobial stewardship, it's not just a problem within human health, but we also need to ensure that we're doing things in the animal and the environmental health worlds. So thank you very much for paying attention. If you have questions about this presentation, you can email us at antimicrobial.stewardship@saskhealthauthority.ca. You can also scan the QR code to visit our website, which is listed there, and find all of our contact information there. Thank you very much.